Hello everyone. Today I would like to speak about bridging the gap between classical and Bayesian notions of robust error models with an application in bioinformatics. So to start with biological motivation, we typically observe over-dispersed data and outlier in microarray or SNP array analyses, and therefore question was asked, how well fitting are these Gaussian models, which are typically applied by packages such as the Bayer conductor package Lemma, which is an empirical base package for linear analysis of microarrays. Would using a more widely dispersed distribution as likelihood better fit this data? And if it does, which impact does the model choice have on biological interpretation of results? As well as, if we find all this, can we use over-dispersed behavior as a means for identifying possible problems in the behavior of certain genes or with the quality of experiments or certain arrays provided here. The statistical motivation of this method is that we collected this genetic data of microarrays, SNP arrays, and we know that they pose several statistical challenges for the data analysis. Typically, this is a scenario where we have a lot less observations than parameters involved, as the number of samples which are typically taken here are a few hundreds to 1,000, whereas the number of parameters here is the number of genes which are looked at, which is typically several thousands or tens of thousands even. Therefore, there are specific algorithmic approaches which we need, and these are typically involving Bayesian methods, as those are able to pool information in a sense through hierarchical bias. Also, there is the noise behavior. As I said before, we know it's over-dispersed. We know it's often non-Gaussian. However, there's no clear knowledge of any systematics of this behavior. Bioinformatics research has moved on from this statistical questioning of array analysis and gone to next generation sequences, whereas these array analyses are typically applied now in medical treatment and diagnostics, and therefore still the challenges remain from the days of old, that this is a big data analysis which has its computational challenges, but also the problem that parallelization is hard to make feasible, not only because MCMC is inherently serial in its nature, but also because we cannot just split up different gene subsets to create smaller covariance structures, as the genes might all be in some way correlated with one another and influence one another. So I originally said that this was meant to build a bridge between Bayesian ideas of robustness and model selection and classical sense of when something is robust. And therefore, I would like to introduce you to the notion of likelihood robustness. Global robustness compares the range of estimates when varying a given set of possible likelihoods. So we are not looking at being robust with respect to certain outlying values, as is the classical notion of being robust, but we look at the whole model being robust with respect to the choice of likelihood which we have taken. Following Schreier-Malkummer, we define a finite set of possible likelihood functions, and we wish to select the optimal one in the classical notion of Bayesian model selection, choosing the one with the highest a posteriori probability of the model given the data. So how do we wish to approach this? We want to provide a general idea that either we use a set of Gaussian students' t-distribution as error models, and we select the single optimal likelihood function from this, or that we build a finite mixture model of Gaussian and students' t-distribution components as an error model for scenarios which would typically have a Gaussian distribution of its errors, such as linear regression or analysis variance models, but also linear discriminant analysis or stochastic processes with Gaussian errors, as well as any other type of models with Gaussian error models which you could think of. So to give you an idea of the specific setting of this, the microarray context model, color-coded in blue, we find the parameters which are of biological interest. This includes the indicator of differential expression, as well as the mean expression of the genes. And on the other hand, color-coded in red, we find the noise model part, which is what we are interested here when we talk about being robust with respect to the choice of the likelihood function. So we have these phi mg's, these rescaling factors, which will rescale our noise from Gaussian to t-distribution. We will talk about it in a few minutes. And also, of course, as we are talking about t-distributions, the degrees of freedom of these t-distributions as a parameter. So to give you an idea, we are looking at an ANOVA model, which for its biological purpose looks at either a gene being differentially expressed or not differentially expressed. And based on this being 0, 1, we will either have a multivariate Gaussian noise or a univariate Gaussian noise here, with the covariance structure making the main difference here. So we compare these two hypotheses against one another. And the other part here is that we wish to rescale the precision of the Gaussian errors, which we've seen before, through parameters, these phi mg's, which in itself are based on a gamma distribution, which includes the degrees of freedom of the t-distribution as its parameter for the scaling. Also, this whole setting therefore provides us with a bivariate normal gamma distribution with the data, the expression data, following a normal where the rescaling of its precision is happening here. Integrating out these rescaling parameters will marginally provide us with a t-distribution for these expressions. Therefore, this is the trick for allowing us to stay within the normal setting by just adding these rescaling factors. So as auxiliary variables, these allow us to create a finite set of model which includes different t-distributions as well as the normal distribution. And by just switching on and off these rescaling parameters, we can change between these different models. The possible noise models, therefore, based on the choice of these degrees of freedom, include the Cauchy distribution for nu equal to 1, different types of student T distribution with various degrees of freedom, as well as the normal distribution, which would be the limiting distribution for nu going to infinity. However, we will cut this off at a maximum value of nu and not just let it converge towards normality, but directly jump to normality. I said that our purpose was to look at quality control and detection of outlying arrays or gene clusters. And we will look at this by looking at the base factor of the T-ness. So how plausible for a certain gene, or you might also, instead of G, have here an N for a certain array, it is to have a certain degree of freedom or degrees of freedom away from normality, as opposed to choosing a normal distribution prior likelihood. And therefore, with this base factor, we wish to test for an equal distribution of over-dispersed values based on a multinomial assumption, which leads us to a Dirichlet distribution for the relative amount of over-dispersed values on the different arrays. Based on this, we can calculate the Savage density ratio, and with given choice for these indications for the Gaussian scenario, we go into this hypothesis testing in this case. So as for the results for microarray analysis, we looked at 14 different data sets, and we found that T distributions with very low degrees of freedom are preferred in all of these, with the posterior mean degrees of freedom ranging from 1 to 4, which is very far away from Gaussian likelihood distribution. Also, we found that the choice of likelihood matters. The list of genes and gene ontology terms generated by these two models differ significantly. And when, instead of just using or making the model choose either student T or Gaussian noise, we allowed it to obtain a mixture of Gaussian and T distributions. We still found that with few interpretable components, 2 to 3, we still have no Gaussian components showing up, and therefore discarded the idea that 85% of the data are Gaussian anyways, and just 15% are off. There's far more off than just 15%. So here again, you can see for these different type of data sets, the differing number of genes as to the common number of genes. And in a bioinformatical setting, depending on those, the gene ontology terms related to the experiment, which is the actual genetic pathways which are activated here. And we can see that this was which is the outcome of interest for biologists. Here we have a very large impact. In almost all cases, there's a huge number of differences as compared to the number of common gene ontology terms between those analyses. And therefore, choice of noise model matters. In order to show you that this is not dependent on whether we only have a few genes being differentially expressed, but also for a large amount of genes being differentially expressed, both scenarios are included here, and in both cases we find similar results that this makes a huge impact. Lastly, I said that we want to look at this for array quality control. So therefore, we created here two scenarios with various types of noise model, creating the setting of the likelihoods here. So we mixed T10, T4 with T10 distributions for a 40 to 50% ratio. The rest is Gaussian, and also T10 with T1. So here are the most widely dispersed ones. Here are the nicer behaved ones. And we have two different scenarios of these simulations. On the one hand, there's an equal distribution of extreme Bayes factors among all arrays. So basically, on all the arrays, something happens that some genes are more widely dispersed than others, but this is due to experimental settings, due to the biological behavior in the background. In the second scenario, we used 75% of the genes which contained the most extreme 10% of the Bayes factors accumulated on a single array, and the rest, the 25%, were left equally distributed among the others. This is a very extreme setting, but we want to see whether this actually is possible for a quality control issue. We see that the Bayes factors immediately go down. So in this case, the plausibility of having an equally dispersed Gaussian, non-Gaussian scenario is highly unlikely, and we find that something is going on a specific array, allowing us to look more deeply into this, to look for quality issue for this specific experiment. So as a conclusion, we used Gaussian instead of the better fitting heavy-tailed noise models, and found that those can lead to erroneous results and conclusions. And we therefore want to argue for replacing these Gaussian models by either something non-parametric, which is typically limited by the sample size for Bayesian as well as non-Bayesian non-parametric methods, or choose something more robust, either heavy-tailed and or skewed methods of likelihood adaptations, or allowing a combination of classical Gaussian components with such robust heavy-tailed components, and differentiate between these scenarios, and therefore allow for even a quality interpretation linked to these very heavy-tailed components. The future work here is that we wish to generalize this to skewed normal or skewed T distributions in order to not just cover the heavy tail effect of wide dispersion, but also the skewness effect of the error model, and still be able to cover this with only a very small number of components, heavy-tailed components, as well as a Bayes Gaussian component. So thank you very much for your attention, and hope to talk to you in one of the sessions or the poster session to chat about new ideas. Thank you very much.